I'm going to be walking you through example queries. Static screenshots, so you're not going to get the full idea of the function. We're going to do the live ones later, but this is to get you walked through. And this will also answer the question that you asked about. One of the most common queries performed by people coming to the database is taking a peptide and searching for that in the database. So I'm going to walk you through doing that right here. The first example is, say I want to search for this epitope here. This happens to be a flu epitope. Those of you who aren't chapter and verse on epitope sequences. So you will come on the home page to the epitope search box or pane, whatever you want to call it, and you will type it in. What's that for? Copy and paste, which is what we mostly do. That takes a tremendous amount of time. And then click search. What this search that I'm going to show you highlights a few things. It highlights the sequence search. We will also then show you homology. So we'll show you how you can use this tool right here to search for homologous sequences. We'll also, again, review the results table and what that looks like, and showing this summary. I'll show you how to filter on particular epitopes. We'll show you how to drill down, and I will show you data details, which is the end of drilling all the way down into the context that we've captured with that particular epitope in that particular scenario. So you run that search, you type in the sequence of interest, and you hit search, and the results page comes up. And what you'll see here is 5 structures matching that exactly, because the default is an exact match of that sequence. You'll see that some of these have this little bit of annotation here. This means that there's been some main sequence modification, some kind of modification that's been done to it, but the amino acid sequence is identical. You'll see that this is the antigen that it came from is NP, and this is from influenza virus. They are, of course, one antigen because it's all NP. There are 312 assays, well studied, and 116 references. This data comes from 116 references. So you can click through the tabs to look at the data, and because we know that all NP, I'm going to click on the Assay tab. When you click on the Assay tab, you'll get a couple of more possibilities here. So within the Assay tab, how much T cell data versus B cell data versus MHC-like evolution data. So here you can see, I have to squint, that there's in parentheses the total number of assays, T cell assays, total number of B, total number of MHC-like. So we see 265 T cell assays, 47 MHC-like evolution assays. You can use these columns here to sort, but also just here we'll take time to familiarize ourselves with this data, this table that is, again, summarizing the data, but of course from all of these summarized tables you can drill down into the exact specific scenario or specific assay. In this place you can click on this assay ID and drill down. You can see that the reference information is summarized, epitope information, the host, the immunization process, whether it was a natural infection or whether it was an administrative thing. In fact, in this case, it's a mouse, so there was an administrative organism. The antigen in the assay, in this case, is the epitope. Here we see, as people asked before, the restriction is here also, and then the assay and whether it was positive or negative. If you unclick positive only, you will see negative data here too when you run the query, so you would see that here. All right. So if you click on the reference tab, again, I'm just familiarizing you with what it looks like when you walk through these things. You'll have a reference ID. This is a PubMed ID that links to PubMed. The author list, the title of the paper, and the abstract, and the date. And this is from most recent down. It'll show you the most recent first. So a really unique feature that is a part of the new results page, so I fall backwards, is that you can remember that initial list of 5 structures. If you're just interested in the first one, you can click here on this filter, and I've highlighted, made it huge here, as you can see. If you click on this filter, it says, tell me just the information for that particular epitope. So as soon as you do that, these will reset, representing the data for just that particular epitope. So then you can look that You've lessened the assays a little bit and lessened, but most of the data is for that naturally occurring. Oh my goodness, I'm going crazy. There we go. Sorry. All right. So that's a neat feature. You can also lock in on just the antigen, and you also can lock in or filter, I should say, on the organism. So now, alternatively, something you might want to do is say you're interested in homologous peptides. There's a lot of data in there, and a lot of times people will type in an exact sequence and they'll get nothing, and they'll get dispersion things. But what you can do is you can look at different levels of homology. So the default is exact match. So say we do that. So say we type in the same epitope or copy and paste and say, show me everything that's in the database that is 70% homologous to that. And hit go. What you'll see now is there's now 160 epitopes that are 70% homologous to that original one that you typed in. So you'll see, again, we'll look through this. You'll see that these are the different sequences that are homologous. Some are longer peptides with homology. Some are just one amino acid difference, up to 70%. What you'll see, this you might ask yourself, well, why is this blank? Why does this not say NP? These are analogs. A lot of times people study synthetic analogs of natural occurring peptide because it defines better or that sort of thing. That's what this data is. So say you're interested in that, and you say, oh, okay, well, I'm interested in this particular one because I look over, scanning over here in these columns here. Well, there's been studies. Look, there's 2 papers and 4 different assays. So let me lock on that. So if I click on that and filter for just that data, you'll see that it'll show that there's a total of 4 assays for that, which is in that column. There, 2 of them are T-cell assays, and 2 of them are probably MHC binding assays, if I had to guess. That's a very common assay. And this is a summary of those 2 things. So now I'm going to show you to drill down to look actually into the data that is represented by this summary here. I'll click on that. When I do that, I will see the entire detailed context that we've captured for this one instance, this one assay for this particular epitope, and I'll walk you through this slowly. At the very top, the information is about the paper. So the abstract, the authors, links to PubMed, anything that's blue links. Down below is, as you scroll down, because this is a long thing that you will scroll down, you will see the next bit of information is the epitope. And because I clicked on the analog, this is the analog here, and it says this epitope is an analog of that naturally occurring one from NP, from influenza. If you continue to scroll down, you then see the T-cell assay information here. This is what I was alluding to earlier. We have these assay immunization comment fields, which are free text fields, similar down here. This is the kind of area where we can put details that are conveyed by the author in the methods and materials, or in the conclusions. We can write things in there, so those are very rich of information right there. There's the host, there's the immunogen. We've talked about immunogen, we talked about antigen. In this case, there is in vitro restimulation. That's there. That's information. This was the type of assay used, whether it was positive, negative, and continue to scroll down. There's the effector cells right here. If the author said that these were CD4s and CD8s, it would be right there, and that would show. It shows you the antigen presenting cells, the MHC restriction, again, if it's recorded and defined in the assay, and then the assay comments are down here. So you can see there's a tremendous amount of detail. These are not always all filled out. It just depends on what detail we get. If we have the detail in the paper, we will capture as much as humanly possible. Sure.
Going back to the slides, in the class search.
Yep.
So back for.
Okay.
So these all came up with nuclear protein.
Yep.
If this epitope were present, a different protein show up,
yeah,
or a different organism.
Yeah.
And is this linked to the NCPI blast database, or separate? Are you only blasting against your database, or blasting against the entire universe?
We are blasting this blast. That's a good question. I'll repeat the question. When we are performing this homology blast, we are only blasting what's in the database.
Your database.
Our database, yes, this database, the IEDB. It's blasting all the other epitope data.
So it's not blasting the protein.
It's blasting to, no, it's blasting to the epitope.
So if you want to blast your peptide against the protein, you go to the IEDB.
Yes. Yeah.
It's first thing.
Yeah, yeah, that's, and we do that all the time. We have both of them open most of the time, two screens, and most of them open. The other neat thing you can do with that is you can actually copy and paste an entire amino acid sequence and paste it into there. Search for epitopes.
Right.
Yeah, yeah, no, that's good clarification, yeah. Yeah. Debbie, later in the afternoon after lunch, Debbie is going to be explaining in great detail. Yeah, we have the ability to do that, so people do that all the time. Question. All right, let's see. Okay, so the second example. Yeah, sure, absolutely.
So, yeah.
Yes.
All of them cannot be epitopes that move outside. But is there a way that we can predict or have a confidence? I mean, how confidently we can think that line
An epitope.
on what chemistry?
Yeah, so I guess the question is, if you're looking for homology and you're doing that using NCBI, doing blast search and alignment, NCBI, and you find regions that are homologous, and you want to see if those regions, is there any likelihood that those regions have been tested in any way, shape, or form? Yeah, you can actually do that. Cut and paste that region and do a search. You can do an exact one. I would say start at 70. Yes, absolutely.
Right.
Put it in the epitope.
No, it would have already given us if it is yes. It's not in here, but we are going back and having signs.
Well, what I'm saying is you might find homologous. You might find information in there that's not exactly what you're looking for, but it might be homologous. And we've done this before in some of the meta-analysis that we've done. We've been able to look and see the data that you're looking for may not have been done yet, but you might be able to find antigens that have been tested with regions that are homologous and find other epitopes. So you can look at it at the antigen level, for example. You also might be able to find epitopes that have been tested that aren't exact, but are perhaps homologous. Maybe they were longer, maybe they tested 15 years that contain the region that you're looking at. That makes sense? And we can look at examples later, too. Yeah, tomorrow we're doing predictions, and then I'll
We see only based on alignment, whether they are actually MSC yes, the percentage of identity is very low, but it may be relevant exactly.
Yeah. But if you can run a prediction on that, but that is again just a prediction based on the sequence. All right. Okay. So the next example. Some of the sorry for the people who are about some of the questions become complex, so repeating them all is a little difficult. So I apologize for that. All right. So say you want to look for all T-cell epitopes that have been associated with treatment of allergy. That's using normal language, but let's break this down into elements of how you want to query for it. So T-cell assays, here we go. T-cell assays is pretty straightforward. So you're looking for T-cell assays, so you'd click here and try the term treatment. In this case, because we're so good at having, or try to be so good at having synonyms, you get the synonym coming up for decreased disease symptoms after treatment. So you can, that is now selected and that is chosen. And then scrolling down on the results page, you can then highlight allergic disease. So now you've chosen T-cell response, specifically treatment assays, and specifically for allergic disease. Now, if you're interested in a particular allergy, you could then further specify this, but I'm just doing this as an example. Then click search. So what you find here on the results page is 77 epitopes that have been associated with treatment of allergies in all hosts. So I didn't specify hosts, this is all epitopes. And you can see some of them are peptidic, some of them are non-peptidic. You can then click on the antigen tab to get a quick look at what's there. And you can see, if you look down this list, pollen, dust mite, bee stings, grass, fungus. You can then click you can use the filter to filter on a particular allergen. You can also do that at this level. The organism, if you're interested in Bermuda grass, you can do that. And also, as we were shown with the immunobrowser, you can map all of these to the reference antigen here. If you click on the assay tab, again, you see the same summary table of all the different assays that were performed, going down, type of assay. You can use, these are all sortable, so you can sort, so you can look at humans. You can quickly click here and sort, look at humans, guinea pig, mouse, whatever happens to be there. And I guess that's the end of my examples. Now Nima's going to take you through some examples. Do you have any questions on the two that I performed? I feel like I'm not coming. Are you still hearing me okay?
So I'm just going to kind of continue that theme and present a couple of example searches sort of framed in a form of questions that an immunologist might ask him or herself. So first one is asking, how would we find antibodies that recognize continuous linear peptides and have been shown to be neutralizing against influenza A virus? So obviously we're doing a B-cell search here, and what I kind of want to hopefully exploit with these queries that are going to be done on screenshots is the fact that some of these queries can be done exclusively on the home page. Some of them require a preliminary home page search and then a further refinement on the second page. So these arrows represent basically the limitations that we can do, or the definitions that we can start with on the search panes to kind of get the query at least started. So you can see I've defined linear epitopes. Using the kind of auto-find feature, you can use the organism finder to type in flu or influenza, and in this case I've chosen influenza A virus to keep it broad. Humans is my host, and I've unselected T and MHC to narrow it to B-cell results only. And so by clicking search down here, you'll see the common page now. I apologize that it's an eye chart. It's very small, but you're familiar now, I think, by having seen this a few times, that you get a tab, essentially organized summary. And in this particular case, you see that we have 105 epitopes retrieved. Now, we've kind of answered our question except for one thing, and that is that these are all B-cell epitopes defined in flu for human hosts, but they are not necessarily B-cell epitopes that have been shown to be neutralizing. And that was the key piece of my query that I was unable to put on the home page. Nevertheless, we can refine this query down here in the assay panel. And so by clicking there or mousing over it, actually, you are able to expand the trees or the finder views that you've seen us several times now. And by going into the B-cell tree view, what you can do is actually, by free text, which is one route, type in the name of the assay or a keyword of the assay in which you have interest. So in this case, I put in the word neutralization, and I've clicked find. And then the recurring theme of summary or hits coming up in tables is shown down here, where in this case you do get a very narrow result of one particular assay, which we call officially neutralization/biological activity. And so I can highlight in tree and show that in the kind of relational organization of our assays, that assay is found there. So you can also have just navigated the tree if you'd like to kind of see what's available. But in either case, if you click that, you'll get that green check, and it will populate the current selection box up here, after which clicking apply will lead you back to the home page where that assay will now be selected. Now, what I've shown you here is a shortcut to what I just did in 5 steps. You could have just began typing neutralization in that box, and you see that the one and only result that comes up is that same assay. And then by selecting it, you don't even have to open up the tree view. You can just shortcut straight to it. However, you still will need to apply that refinement by clicking search. So after you select that, you'll see that the search will light up green again. One common pitfall is that people will apply that neutralization and wonder why these numbers didn't update, because the system is waiting for you to say execute this, because you can update multiple parameters, right, before. So if I go ahead and click that search, excuse me again. If I click that search, you see that I have now refined my epitopes down to 5. And as Carrie has been demonstrating, one can then kind of survey the different tabs and reveal that all 5 of those epitopes come from hemagglutinin, as might be expected. And you can furthermore explore the assays tab and namely go down to the B cell assays. Notice these 2 are 0 because we excluded them, but this particular B cell assay shows you a number of, again, in summary view, as Carrie pointed out, of each assay and from which reference it comes. And if I click any one of these assay IDs, which is a unique ID that we assign when that assay is input into the database, you will be able to go down into the real bread and butter data of the IEDB, which Carrie showed you before, so I won't belabor it, but it's essentially every single data point relating to that epitope. This is basically what, this is the job security for us curators. We manually extract every single one of these data points, right? Okay, so I put this slide in to just kind of point out the fact that if I would have done that exact same query that I just did with one minor change, that is, instead of clicking linear, if I change my epitope definition to discontinuous, you in all likelihood will get far more results. We do capture, as been mentioned, discontinuous B-cell antibody epitopes, and I don't know the numbers, but I would venture a guess that probably somewhere around 50% of our monoclonal data is discontinuous or somewhere, it's a good chunk, maybe not 50%, but there's a lot of data out there, so please keep that in mind if you're a B-cell person, that definition has to be made by you, otherwise you will only see the continuous epitopes. All right, and then I'll just briefly take you through a similar T-cell search, again an infectious disease-related search. But if the quest now is to find Mycobacterium tuberculosis epitopes that might have been used to develop H2KD tetramers to detect T cells in biopsy mice, so very often we get this question of there's that NIH resource, the tetramer resource, but a lot of people turn to us to see whether people have developed tetramer technology for epitopes because they simply want a reagent and want to know whether such thing has been done. So again, this is a matter of just dissecting this question into pieces and filling in the blanks on the homepage search, which I've done here with the organism, the host, etc. You can see here I've just limited it down to a MHC class 1. The reason being there's no ability on the homepage search, I was on this interface, to define a specific allele. But I've taken the query as far as I can, and by clicking search, again, I have a query result which is somewhat close to what I desired, but again, artificially inflated because these are not just KD epitopes. These are all class 1s that are defined in rodents. So again, I can go down here. Now I have, pardon me, I now have the luxury of opening up that finder, if I so choose, and defining AD with the allele finder. But what I wanted to point out, kind of like a little bit of a trick or a habit you might find yourself picking up after a while, is to just kind of scour the data and explore it and see what's out there, because some of the filtering capabilities that we've added in that Carrie demonstrated might come in handy and are actually quicker sometimes than using the search interface. So if you here were to follow that red arrow and click on the Assays tab, you would right away notice that, and I've no way sorted this data or filtered it beyond that, but right away I noticed that in my MHC restriction column that there are a few instances of KD epitopes already there, and my original target was KD epitopes. So one could just click that filter and immediately delimit or filter out those results to KD epitopes. And then by definition, you're going to get all your hosts in mouse anyway. You wouldn't have to go and worry about changing the rodents to delimit it further. So by doing that, you can see I've narrowed the results significantly. And again, just another example of how these filters can sort of come in handy. If you go to the antigens, you can see that in this particular case, those KD epitopes have pretty broad coverage in terms of antigens. I don't know how many there have, maybe a dozen different antigens there are represented. And we also have an assay in the assay finder that's specifically called tetramer assay. So if you wanted to be 100% certain that these were used for tetramers, you would, of course, go into the assay finder and define tetramer assay. And I don't believe I showed that step, but wanted to mention it. We'll have a lot more examples as well to go with that. Okay, so taking you a little bit over the lunch cutoff, are we going to just call it break now, or any quick questions or anything before we head out to lunch? Okay, so right out this door to the left,